14 to 17,000 years ago, across sites in Germany, France, and Switzerland, the skeletal structure of certain canids began to change. Their snouts shortened, their jaws widened, and their overall size decreased. These remains stand as the earliest morphologically identifiable dogs on the archaeological record. This physical timeline creates an immediate geographical problem. While the oldest fossil evidence of domestication lies buried in Europe, modern genetic analyses point consistently east, suggesting dogs share their strongest affinities with wolves from eastern Eurasia. For decades, the fossil record alone lacked the resolution to settle this contradiction. Because morphologically ambiguous bones cannot yield a complete evolutionary map, researchers maintained a highly plausible hypothesis that wolves were domesticated twice, once in the East and independently in the West. Settling the debate over a separate European domestication depends on extracting uncorrupted nuclear DNA from bones that have been decaying in the Earth for 14 millennia. At the Kessler-Loch cave site in Switzerland, researchers uncovered a canid specimen dating back 14,200 years. The physical reality of a specimen this old is that the original biological material has heavily degraded. In pre-Neolithic remains like these, over 99% of the genetic material extracted does not belong to the animal. It belongs to environmental soil microbes that colonized the bone over thousands of years. Standard shotgun sequencing on these samples yields less than 1% endogenous DNA, making the process both expensive and inefficient. A team led by Anders Bergstrom set out to bypass this microbial noise entirely. Their objective was to isolate that fractional percentage of true canid nuclear DNA from 216 ancient remains scattered across Europe. Breaking through this biomolecular bottleneck is the prerequisite to reading the population structure of Ice Age dogs. Without it, the genetic history remains invisible. To solve the degradation problem, the team deployed a technique called hybridization capture. Rather than sequencing every piece of DNA in the sample, researchers synthesized targeted genetic probes that bind exclusively to specific known canid sequences, allowing the surrounding microbial DNA to be washed away. To avoid biasing the results toward any specific modern dog breed, these probes were designed using a genetic outgroup, a modern coyote. They specifically targeted over 486,000 single nucleotide variants. A single nucleotide variant, or SNV, is a specific position in the genome where a single letter of the DNA code differs among individuals. By focusing purely on these pre-identified genetic markers, the capture protocol resulted in a 10 to 100-fold enrichment in usable endogenous canid DNA. This scatter plot visualizes the results using an F3 statistic. The F3 statistic is a mathematical tool that calculates the amount of shared genetic drift between two populations relative to an outgroup. The dotted threshold line precisely separates the genetic drift signatures characteristic of wolves from those of dogs. The algorithm is calibrated against a modern baseline to establish the specific axis of shared genetic drift that identifies a specimen as a domesticated dog. In this study, the modern African Basenji was used as that crucial reference point to anchor the statistical test. Evaluated against this strict metric, the 14,200-year-old Kessler-Loch specimen confidently crosses the threshold. Mathematical certainty replaces morphological guesses, officially validating Kessler-Loch as the oldest genetically confirmed dog. With the European Paleolithic dog validated, the core evolutionary question remains: did this animal arise from a local, independent domestication of European wolves? This chart maps the genetic relationships between canids using a principal component analysis, a statistical technique simplifying complex datasets into visual clusters. Here, modern wolf diversity is mapped along a clear east-west geographic line. When the genetic data of pre-Neolithic European dogs are projected, they do not align with Western European wolves. Instead, they cluster strictly on the eastern side, alongside Arctic and Asian dogs. To formalize this observation, researchers utilized the QP-ADM statistical framework. The QP-ADM framework is an algorithm that tests whether a target population's ancestry can be modeled mathematically as a mixture of specific source populations. These bar charts compare the viability of two distinct evolutionary models. The first tests if ancient European dogs derive 100% of their ancestry from an eastern Siberian progenitor. The second tests a dual-source model, requiring an additional genetic contribution from a western Syrian wolf. The single-source eastern model fits perfectly. The ancestry of all analyzed pre-Neolithic European dogs can be entirely explained by the eastern progenitor, with zero mathematical requirement for a western wolf contribution. The independent European domestication hypothesis is statistically eliminated. The earliest known dogs in Europe derived from a single eastern Eurasian wolf population, migrating westward after domestication had already occurred. Understanding their origin from the east immediately raises a secondary issue of internal evolution. Once these eastern progenitors settled in Europe, how did they structure their populations over the ensuing millennia? This bar plot tracks exploratory genetic affinities using admixture clustering, a software tool that assigns fractions of an individual's genome to inferred ancestral populations, visualizing them as different colors. Across the timeline, a unified genetic component emerges, dominating later European Mesolithic dogs. Yet, the 14,200-year-old Kessler-Lock dog sits outside this profile. Its data column shows highly divergent ancestry, lacking the uniformity seen in dogs that lived millennia later. By directly contrasting Kessler-Lock's shared genetic drift against other global populations, researchers traced where this missing affinity leads. The analysis reveals that the Kessler-Lock dog shares an early, highly specific genetic drift with an 11,400-year-old dog excavated from Bonkuklu Hoyuk in Anatolia. This specific connection breaks the linear genetic relationships observed in later European dog populations. This divergent profile demonstrates that deep genetic differentiation already existed among dogs 14,200 years ago. It pushes the timeline of their global diversification deep into the Pleistocene, long before they appear in the Swiss cave. This established Mesolithic dog population eventually encountered the next major chronological event in European history, the Neolithic agricultural revolution, driven by the mass migration of farmers from Southwest Asia. The human genetic baseline during this transition is well documented. As early agricultural societies expanded westward, the incoming farmers genetically replaced 70 to 80% of the indigenous European hunter-gatherer populations. The demographic question then applies to their animals. Did the massive influx of Southwest Asian farmers' dogs similarly eradicate the indigenous Mesolithic European dogs? This map illustrates the post-Neolithic dog admixture proportions across different European geographies, displaying the ratio of incoming Southwest Asian dog ancestry against the indigenous Mesolithic lines. The data reveals extreme regional variance. In Southern Europe, a dog from Neolithic Greece shows 66% Southwest Asian ancestry. Further north, a late Neolithic dog in Germany sits at 53%. In Denmark and Sweden, incoming agricultural ancestry drops to nearly zero. The dogs do not mirror the human demographic history. The incoming farmers did not wipe out the local canid populations. Instead, they actively incorporated the indigenous hunter-gatherer dogs into their agricultural packs. This branching diagram shows a phylogenetic tree synthesized entirely from the newly extracted nuclear evidence. A phylogenetic tree is a diagram showing the evolutionary relationships and common ancestry among various populations. Here, all global dog lineages trace back to a single eastern wolf progenitor. The European branch splits off remarkably early, cementing the deep population structure proven by the 14,200-year-old Kessler-Lock sample. When the timeline reaches the Neolithic agricultural transition, the European branch does not terminate. Instead, the incoming Southwest Asian lineage merges into it, creating a heavily braided genetic line rather than an override. For decades, highly degraded bone fragments left this history open to interpretation. By utilizing genome-wide capture technology to filter out microbial noise, researchers finally resolved the morphological ambiguity with concrete molecular data. The survival and integration of the Mesolithic dog lineage represents a distinct demographic anomaly in the history of Holocene expansions. This ancient admixture data directly informs the genetics of the animals living today. Modern European dogs still trace roughly half of their genome directly back to these pre-agricultural indigenous Ice Age populations. This stands in stark contrast to the human genome, where the genetic legacy of the European hunter-gatherers was largely lost. Humanity's first domesticated animal survived the greatest human demographic shift in history by integrating into the newly arrived agricultural societies.